This week's lecture material is based on information from Chapter 19 in your textbook. We'll be looking at the physiology of blood. A reminder that the PDF format of these files is posted in your Blackboard shell, and you want to make sure that you're looking at those slides as well, because there'll be more slides in that presentation compared to what you're going to see on this voiceover PowerPoint presentation. It's important to know that blood has many different important functions in the body. One of the main functions is as a form of or a vehicle for transport, transporting oxygen from the lungs to the tissues, transporting carbon dioxide from the tissues back up to the lungs so that we can breathe it out, transporting nutrients, so when we consume foods that are broken down into their basic building blocks in the digestive system, those nutrients enter the bloodstream and then are carried to their target sites. As well, blood will function to transport hormones. Hormones are chemical messengers that the body uses to communicate between tissues and cells and organs. Another important function of blood is to regulate pH. We learned in our last lecture that there's a buffering proteins that are present in red blood cells and as well in the plasma of blood. There are proteins that help to buffer the pH so that we maintain our homeostatic levels of 7.35 to 7.45 pH in the body fluids. Another important function is the temperature regulation. So, for example, if we're overheating, if we're getting too warm, the body can shunt blood to the surface, to our skin, so that we can lose heat from that blood to the environment around us and help to cool us. And conversely, if we're feeling we're too cold, our body will shunt blood away from the surface of our skin towards the core to try to maintain or to retain the heat. Another important function is protection, so against foreign antigens, viruses, bacteria, etc. that might enter our body. The different antibodies that we have and the white blood cells that we'll be looking at in this lecture as well are an important part of our immune system. So these cells travel within the blood itself. And the final function that we're looking at will be clot formation. So there are proteins and cells within, or chemical mediators within the blood that function to create clots, so that if we were to have a cut in a blood vessel, that would initiate a cascading series of events that will result in the formation of a clot so that we don't bleed to death from the smallest cut. Let's look at the composition of blood now. There's two major categories of stuff that makes up blood, formed elements and plasma. Blood is a connective tissue. It's a type of connective tissue, and just like all connective tissues, there's a matrix component, and then there's a cellular component to the connective tissue. So the matrix component of blood is plasma, and that makes up 55% of the composition of blood. And plasma is composed primarily of water. About 90% of plasma is water. There'll be proteins in that watery matrix, as well as other solutes that we'll look at. And then we have the formed elements, and the formed elements that comprise about 45% of blood are the cells. So the three types of cells that we have in blood are the red blood cells, which are also known as erythrocytes. We have white blood cells, which are known as leukocytes, and then we have platelets, and platelets are also known as thrombocytes. The cells that make up the formed elements of blood then are red blood cells, also known as erythrocytes. Red blood cells will have the protein hemoglobin inside them. Hemoglobin is the structure that will bind oxygen and carbon dioxide so that we can transport these gases in blood. The white blood cells, also known as leukocytes, are divided up into granulocytes and agranulocytes. So this refers to when we stain these cells to be able to look at them under a light microscope, the granulocyte will show large granules in the cytoplasm, and as well these cells will have multi-lobed nuclei. three types of granulocytes. We have neutrophils, eosinophils, and basophils. And then the agranulocytes, when these cells are stained, will not illustrate large granules. They have small granules in the cytoplasm, and their nuclei are not lobed. So, and we have two types of cells that are agranulocytes. We have lymphocytes and monocytes. All of these white blood cells, all these leukocytes, have a major function as a component of our immune system, so it helps to identify and destroy foreign antigens that may be in our blood, such as bacteria, viruses, and as well they play an important role in the inflammatory reaction in the body's response to injury and trauma. The third type of cell is the platelet, also known as thrombocytes. Platelets are not considered to be real cells or complete cells. They're fragments of a larger cell that have been broken off, and platelets have a very important role in creating a blood clot. So it's necessary for blood to be able to create a clot if there's been trauma, injury to a blood vessel. Platelets are required or the necessary component of creating that clot. This figure is in your textbook, and it's just illustrating again the composition of blood. So in the test tube we see that 55% of blood is composed of plasma, 45% of blood is the formed elements. If we break down plasma, we see that the majority of plasma is water. 91% of plasma is comprised of water. 7% is proteins, and 2% is other solutes. So if we look at the proteins, we have three types of proteins in plasma. The most common is albumin, but we also have globulins and fibrinogen, and we'll look at the functions of these different proteins further on in the PowerPoint. And then when you look at the solutes that are in plasma, it includes the ions like sodium, potassium, chlorine, bicarbonate. Nutrients, so the nutrients that are being transported in blood will be found in the plasma. Waste products of cellular metabolism, acids, etc. will be transported in the blood. The gases, primarily carbon dioxide, most of the carbon dioxide that's in blood is transported within the plasma. And regulatory substances like hormones that we mentioned earlier will be found in the plasma as well. When we looked at the formed element component of blood, remember we have platelets, we have white blood cells, and we have red blood cells. And the white blood cells are further broken down into the neutrophils and lymphocytes, the monocytes, the eosinophils, and the basophils. You do have to know that the neutrophils are by far the most common type of white blood cell, the most common type of leukocyte in blood. If we were to put whole blood into a test tube and then put that test tube into a centrifuge, it will separate the whole blood into these three components. So you will have plasma, and then you'll have the red blood cells, and then you'll have what's known as a buffy coat, which is a thin layer of leukocytes and the platelets. So notice that the buffy coat is only comprising less than 1% of the total volume of whole blood. Hematocrit refers to the percentage of red blood cells by volume in whole blood. So the hematocrit normally should be about 45%. So if someone's having blood work done and their hematocrit is above 45%, it means that they have too many red blood cells in their blood. And if the hematocrit is below 45%, then it can indicate that their body's not making enough red blood cells. The red blood cell count that's done is the number of red blood cells counted in 1 cubic millimeter of blood. If we look at the structure of red blood cells, of erythrocytes, first of all, we know that red blood cells have a protein. They contain the protein called hemoglobin, and this is the structure that will bind oxygen within the red blood cell. Red blood cells don't have a nucleus, so they're anucleate or organelles. And also the structure, the shape of the red blood cell is very important. It's a biconcave disc, and the reason for this shape is that it provides a very large surface area available for diffusion of gases across its cell membrane. The structure of the cell, of the red blood cell, is such that it's very flexible and deformable. so that when these red blood cells are entering into the capillaries, which are the smallest diameter blood vessels in our body, and that's where the transfer of gases occur. The carbon dioxide and oxygen transfer occurs in the capillaries. And these red blood cells are having to squeeze into these capillaries single file, one at a time, and are deforming to be able to get into these capillaries. And this allows for a very efficient process of diffusion across the cell membranes of the red blood cell and the capillary itself. Oxyhemoglobin versus deoxyhemoglobin. So when hemoglobin has bound to oxygen, it's now called oxyhemoglobin. And when oxygen is released from hemoglobin, it's known as deoxyhemoglobin. If we look at how the respiratory gases, oxygen and carbon dioxide, are transported in blood, if we're going from the lungs to the tissues, so in the lungs the blood picks up oxygen from the air that we breathe in, and we see that 98.5%, 98.5% of oxygen is attached to hemoglobin within the red blood cell. About 1.5% will be dissolved in the plasma. So vast majority of oxygen is attached to the hemoglobin within the red blood cell for transport. Carbon dioxide, on the other hand, so the carbon dioxide is a waste product of cellular metabolism, so that's being formed in the tissues, and we have to get that carbon dioxide out of the tissues into the blood and transport it to the lungs where we can exhale it out. So carbon dioxide, first of all, the important thing to know, you'll definitely get asked this, the majority of carbon dioxide is transported in plasma in the form of bicarbonate ions. So the HCO3 negative is bicarbonate, and the carbon dioxide is within that molecule, that's the form that it's being transported in plasma. 23% of the carbon dioxide will be in combination with hemoglobin. So when carbon dioxide binds to hemoglobin, it's known as carboxyhemoglobin, or the hemoglobin is known as carboxyhemoglobin. And then 7% of carbon dioxide is in plasma in a dissolved form. So the vast majority, 70%, will be transported as bicarbonate ions within plasma. This equation represents the reaction that occurs within the red blood cell. And we've seen this when we were talking about the buffering of acids within blood. But if we're looking at it now in terms of how carbon dioxide is transported in plasma, the series of events that occur, first of all, CO2, carbon dioxide, is made in the tissues as a waste product of cellular metabolism. It then diffuses out of the tissues and into the plasma of the blood capillary, and then the CO2 will enter the red blood cell, and some of it will then bind with hemoglobin to form carboxyhemoglobin, but the majority will enter into this equation. CO2 plus water within the red blood cell will go to form carbonic acid, H2CO3, and this is catalyzed by the enzyme carbonic anhydrase. And then carbonic acid will then dissociate into bicarbonate ions, HCO3 negative, and hydrogen ions. And now the bicarbonate is able to leave the red blood cell. It diffuses out of the red blood cell and into the plasma, and this is the form in which approximately 70% of carbon dioxide is transported in blood as the bicarbonate ion in plasma. We're going to look more closely at the structure of hemoglobin. Remember that this is the protein that will bind oxygen within the red blood cell. It's a pigment, so it gives the red blood cells that red color. It's a structure that will bind very easily and reversibly with oxygen, and each red blood cell will contain approximately 250 million hemoglobin molecules, and each hemoglobin molecule is able to bind 4 oxygen molecules, so we can carry approximately 1 billion oxygen molecules in each red blood cell in the body. If we look at the structure of the hemoglobin molecule, it's broken down into 2 components. We have the globin or protein component, and we have the heme component. The globin component is made up of 4 polypeptide chains, 2 alpha and 2 beta chains, and associated with each of these chains is a heme component. So if you see the image on the right side of the slide, that's representing the heme structure. And we see in the middle of this heme structure that there is an element of iron. Fe is the chemical symbol for iron. And this the heme has to have an iron component to be able to bind reversibly to oxygen. So if we don't have iron, if we don't have enough iron, we can't bind, red blood cells can't carry oxygen effectively. So it can create a type of anemia known as iron deficiency anemia. Most of the iron that's in these heme structures are recycled or is recycled. So when red blood cells are broken down to create new red blood cells, that iron will be recycled for the most part to go into the new formation of the new red blood cells. However, we do lose a little bit of iron each day. We lose a little bit in the urine and feces and in menstrual blood. So there has to be a replenishment of iron stores from our diet. So this is a concern. You'll learn further on in your courses when you learn about anemias that if somebody is very strict vegetarian, iron deficiency anemia is a concern because most of the iron that you get in your diet comes from animal sources. The normal lifespan of a red blood cell is approximately 120 days. And what tends to happen, recall that the structure of the red blood cell is such that it's very flexible, very deformable. And as the red blood cell gets on in its years, gets towards the end of its lifespan, it becomes less flexible, less deformable. So eventually it gets picked up through the as it's going through the spleen and the liver. These are structures that act as very fine filters in the circulatory system, and if these red blood cells are becoming less flexible, less deformable, they end up getting picked up in the spleen, and then macrophages, part of our white blood cell population, are able to break down, destroy these red blood cells. So here we have a macrophage, and we see that the red blood cells these old red blood cells are getting eaten by the macrophage, and the macrophage now will start to break down the red blood cell. So we're going to break it down into two components. We're going to break it down to the globin component, the protein component, and we can now use those amino acids that are made up, that are making up those globin chains, to reuse these amino acids to build other proteins. And then the heme component, remember we want to recycle the iron, so the iron will come off the heme component, and we'll keep that iron in the body to reuse it to build more red blood cells, and we'll see that we make red blood cells in our bone marrow, in our bones. So the iron will go towards rebuilding new red blood cells. And then what's left of the heme component becomes a structure known as bilirubin, and the bilirubin will end up going to the liver, where it's going to be conjugated to create another product that will enter into bile. And bile is the fluid that it's made in the liver and it's stored in the gallbladder, and bile is necessary for digesting fats. When we consume fats, we have to put some bile into the digestive tube to help to emulsify the fats in the watery medium of the digestive tube. So from there, once it's in the bile, the bilirubin will enter into the intestine, so it'll be then excreted through the feces, and then some of it will also go to the kidney and come out in the urine. So this is the bilirubin as a end product of hemoglobin breakdown. If somebody's having problems with the liver, if they have liver issues like cirrhosis, for example, there's a common symptom that you see. Bilirubin is a pigment as well. It's a yellow pigment. And if there's a problem with the liver not able to conjugate, package the bilirubin to get rid of it, you end up getting increasing levels of bilirubin in your blood, and it creates a condition known as jaundice, where you see a yellowing of the skin. The term erythropoiesis refers to the production of red blood cells. The kidney is the major organ that stimulates the process of erythropoiesis. And what initiates this process is the blood entering the kidney if it's not carrying enough oxygen. So it's in a state of what's known as hypoxemia. Whenever you see the suffix emia, it refers to blood. So hypoxemia means there's not enough oxygen in the blood. And the kidney monitors this, and if it sees that this is happening, it will release a hormone called erythropoietin. Erythropoietin will then act on red bone marrow within certain bones of your body to increase red blood cell production. So as the red blood cells are being produced, they're going to be able to carry more oxygen in the blood. And once we reach homeostatic levels of blood oxygen level, that's going to tell the kidney to stop releasing the hormone erythropoietin. To make enough, to make red blood cells, we have to have the elements iron. We already saw that we need iron to make heme, to be able to bind oxygen. But we also require sufficient B-complex vitamins, folic acid, which we get from leafy green vegetables, and B12 as well. And B12 is a concern again for strict vegetarians because B12 only comes from animal products. So there's a concern if you have somebody who is a very strict vegetarian, has no animal products in their diet, that they're not getting B12 from their diet. There's a chance or an increased possibility of developing a B12 deficiency, which can create a type of anemia because you're not building enough red blood cells. We already discussed anemias briefly. You'll do much more detail in different types of anemias in the pathology courses that you'll be taking. But generally, anemia refers to the decreased ability of blood to transport oxygen. And there are many, many different causes of anemia. So this results in a state of tissue hypoxia. So hypoxia refers to a condition where there's not enough oxygen available. Polycythemia is another type of erythrocyte disorder that you'll learn about. And in polycythemia, the red bone marrow is making too many red blood cells for whatever reason, and there's a concern with this because with too many elements, formed elements, too many cells in the blood, it creates a more sluggish blood, so increased viscosity or stickiness to the blood, and this can potentially start to block small blood vessels, the capillaries in the body, and therefore tissues that are being supplied by those blood vessels are not getting enough oxygen or nutrients. We're going to look at white blood cells now. Remember that white blood cells are also known as leukocytes, and the major function of the white blood cells is in defense and immunity. So they're not involved with oxygen or carbon dioxide transfer, so they're not involved with the transfer of the respiratory gases. If we look at what the structure of these cells are like, so as opposed to the red blood cell, white blood cells do have a nucleus. And recall that the granulocytes, which are the white blood cells that have the granules in the cytoplasm, they tend to have a multi-lobe nucleus. And the agranulocytes have a nucleus that's not lobed, but all white blood cells have a nucleus, and they have the normal organelles that we learned about earlier in the course as well. So their function is for protection. They protect against viruses, against bacteria, parasites, tumor cells. They're going to help to remove dead cells, dead tissue, debris, clean up the area after an injury or trauma, clean up the area after the inflammatory process. And this is their major function within the body. Some definitions to learn concerning white blood cells. Leukopoiesis refers to the production of white blood cells. And this occurs also in the red bone marrow. All cells, all blood cells are made in the red bone marrow. So just like red blood cells, white blood cells are also produced in the red bone marrow. And in the case of white blood cells, the production of them will be stimulated by chemical messengers within the body, things like interleukins and colony stimulating factors that will be stimulated by when there's a need to increase the number of white blood cells. For example, if we have something in our body that's foreign that needs to be looked after, destroyed, we'll have these signaling chemicals that are released by cells that will stimulate the production of increased white blood cells. Leukopenia refers to a state of abnormally low white blood cell count. Leukocytosis refers to increased number of white blood cells, and this is what you normally see with processes of infection. When there's an infection going on in the body and they do blood work, you tend to see an elevated white blood cell count. That means that something is stimulating the production of more white blood cells. And leukemia is a disorder, it's a cancer concerning the white blood cells. So it's basically a cancer of white blood cells, and you'll learn more about leukemias in pathologies as well. Characteristics of white blood cells include these three different types of movements: emigration, chemotaxis, and amoeboid. So emigration used to be called diapedesis. The new term is emigration, and this refers to the ability of white blood cells to move into and out of blood vessels. So if there's an injury in an area in the tissue, white blood cells arrive into that area via the bloodstream. They're in the blood vessels, and they can enter into the tissues by basically squeezing out between the pores of the cells that make up the wall of the blood vessels. So this is known as emigration. Chemotaxis, there is a signaling that goes on with, particularly from injured tissue, injured cells will release chemicals that the white blood cells will respond to and will be attracted to that area. That's telling them that there is something here that needs to be addressed. We have an invasion of a foreign antigen, or we have damaged cells, and so these signals are released, and that's what attracts the white blood cells to the area for them to do their work. And then thirdly, we have the amoeboid action of these white blood cells. I'll provide another YouTube video link that will show I believe it's a neutrophil hunting down a bacteria and eventually swallowing it. So once these white blood cells have caught the scent, so to speak, so they're reacting to these chemical signals released by either a foreign cell or damaged cells, they will now hunt or move through the tissues in this amoeboid action by extending out these cytoplasmic extensions until they can catch the foreign antigen, surround it, bring it in, and destroy it. Looking more closely at the granulocytes, neutrophils are by far the most common type of white blood cell, and you need to know that they're the first cell that will appear during an inflammatory reaction. Neutrophils are nonspecific phagocytes, and all white blood cells are phagocytes. So nonspecific means that the neutrophil doesn't care what it's endocytosing. As long as it's identified as foreign or a damaged cell, it's going to endocytose and destroy that material. Eosinophils are associated with the allergic and asthmatic reactions that occur in the body, and are also responsible for destroying parasitic worms that may have entered the body. Basophils are the least common granulocyte, and they're able to leave circulation and go into the tissues. So they have an important role in the inflammatory response and also allergic reactions. They contain granules that have histamine and heparin. Histamine is a very potent vasodilator, so it'll dilate blood vessels and allow fluid to leave the blood vessel and enter into the tissues. And part of the function of that is to try to wall off if there's a foreign antigen bacteria that's entered into the tissue, that these fluid that comes into the tissue will help to prevent it from leaving that area. It walls it off. But it's also responsible for the symptoms that we see during an allergic reaction, with that watery exudate that's pouring out of your nose and your eyes, etc., and that swelling, congestion that you see. It also, these heparin granules that it contains also is a potent anticoagulant, so it doesn't allow blood to clot while these reactions are occurring in that area. Looking now at the agranulocytes, we have two types. We have lymphocytes and we have monocytes. And then if we look at the lymphocytes, this is broken down into T lymphocytes and B lymphocytes. Remember that all white blood cells are produced in the bone marrow. So B lymphocytes are cells that are produced in the bone marrow and also mature in the bone marrow. So B stands for bone. Whereas the T lymphocytes, when they're produced in the bone marrow. When they leave the bone marrow, they're still in an immature, inactive state, and they have to go from the bone marrow to the thymus, another gland in your thoracic cavity that performs what's known as thymic education. It completes the process of building a mature T lymphocyte. So T refers to thymus, the gland that will educate or to complete the process of building a new lymphocyte. So lymphocytes are very closely associated with immunity. The T lymphocytes will act directly against virus and tumor cells to identify and destroy. And the B lymphocytes become what are known as plasma cells, and it's the plasma cells that will create antibodies, which are the structures that respond to, once you've been exposed to a virus, you create these antibodies. So the next time that you're exposed to the same virus, you now have a protection against that virus via these antibodies. Monocytes are the largest blood cell that we have, and they're able to leave the circulation and go into the tissues to become macrophages. And macro means big, phage means to eat, so they're big eaters. Okay, they're big phagocytes. So they're phagocytic against viruses, bacteria, parasites, and they can also present these antigens, these foreign antigens, to lymphocytes so that they can recognize them and either create a memory or able to destroy them as well. The final cell type then that comprises the formed elements of blood are the platelets. These are also known as thrombocytes. Remember that platelets are not complete cells. They're cell fragments that have been pinched off from a mother cell known as a megakaryocyte. That's also in the red bone marrow. And the production in the red bone marrow is stimulated by the hormone thrombopoietin to create these thrombocytes. Remember that platelets have a primary function in preventing blood loss. They can create platelet plugs that will initiate, or it's important in the process of creating a clot that will stop bleeding from an injured blood vessel. So if we look at overall the production of all of the formed elements, all of the blood cells, the red blood cells, the white blood cells, and the platelets, this is known as hematopoiesis. So remember, erythropoiesis refers to the production of red blood cells. Leukopoiesis refers to the production of white blood cells. Thrombopoiesis would be the production of platelets, but hematopoiesis refers to the production of all blood cell lines. All of these blood cells are produced in red bone marrow, and we find red bone marrow in the adult, we find in the axial skeleton. So this includes the shoulder girdles, the pelvic girdle, the ribs, the vertebrae, the sternum, and the proximal epiphyses of the long bones of the humerus and the femurs. And all blood cells are formed from a single stem cell. So from one stem cell known as the hematopoietic stem cell, all of these bloodlines differentiate from this single stem cell. This slide then will illustrate the process of making the different blood cells within the red bone marrow. And remember that all of these bloodlines will originate from one stem cell. So here they're calling it the pluripotent stem cell. We mentioned earlier that it was the hematopoietic stem cell. It's the same thing, and just referring to the fact that from this one type of cell, all of these cell lines, bloodlines, can be created. So we have two major categories of cells. Once we differentiate from this initial hematopoietic stem cell, we can create a myeloid line, and we can create a lymphoid line. And the myeloid line then will end up making the monocytes, the neutrophils, the basophils, the eosinophils, the platelets, and the red blood cells. And the lymphoid stem cell line will then create the T lymphocytes, the B lymphocytes, and also another cell type called the natural killer cell. Remember that the B lymphocyte then can differentiate into a plasma cell, and it's the plasma cell that will respond to the presence of a virus to create antibodies. And notice also that we have the monocyte. Remember that the monocyte can then enter into tissues to become the macrophage, the big eater, the big phagocyte. And to emphasize again, all blood cells, red blood cells, white blood cells, and platelets are produced in the red bone marrow. And we see that the top image illustrates that these are all being initiated from a single stem cell type that will then differentiate into these different bloodlines, and then these blood cells will then leave the bone to enter the circulation. Looking at plasma now, so remember that the majority of plasma is made up of water, 91%. You don't have to know exactly these numbers, but just know that plasma is mostly made up of water. We also have plasma proteins, and we'll look at those different proteins in a second. And then the remainder will be the different gases that we see, the oxygen, carbon dioxide, nitrous oxide that are gases that we see in the plasma. The different ions like sodium, potassium, hydrogen ions, magnesium, chlorine, phosphate, etc. Waste products, so lactic acid, uric acid, bilirubin from the breakdown of red blood cells. These are things that we'll see in the plasma component as well. And regulatory substances, hormones, enzymes. So these are all things that we see in the plasma component of blood. The three types of proteins, the proteins that we see in the plasma are produced in the liver. The most common protein is albumins, and albumins have a very important role in the production or creating osmotic pressure in blood, so which means the attraction of water fluids to the inside of the blood vessels. These proteins provide that osmotic pressure. And they're very important. We learned the buffering action of proteins, so these albumins have an important role in buffering pH within the plasma. And they're also important as transport mechanisms. So both the albumins and globulins have a role in transporting substances that are fat-based in blood. Because remember that fat and water doesn't mix. Blood is primarily a watery medium, so if we're trying to transport, for example, a fat-based hormone, a steroid hormone, or cholesterol, we have to bind it to a protein carrier to be able to transport it in blood. So albumins and globulins also act as these protein transport carriers. Globulins are the second most common type of protein, and besides the transport of fats, they're also involved in creating antibodies. So gamma globulins, for example, are important in creating the antibody structures, also known as immunoglobulins. And then fibrinogen is a very important protein, blood protein that's involved in blood clotting. It helps to form the fibrin threads that will create that blood clot. Hemostasis refers to the stoppage of bleeding. So this is necessary so that we don't bleed to death from the smallest cut. And this involves clotting factors that are present in plasma, in the blood, as well as substances that will be released by platelets and or injured tissue cells when there's damage to that area. And these signaling molecules, these chemicals, will initiate a process whereby eventually we develop a clot that will stop the bleeding in that area. So the three phases of hemostasis. The first phase, the first thing that happens is vasospasm. So when there's injury to a blood vessel or to tissue, the blood vessels in that immediate area will constrict, go into a vasospasm to try to reduce the loss of blood. Secondly, a platelet plug will be formed, so the platelets will mobilize to stick together to create a temporary plug, so that we're again trying to limit blood loss. And then once these different clotting factors are initiated, they will create eventually a blood clot or a coagulation of the blood that will reinforce that platelet plug. And then using the proteins within plasma, the fibrinogen proteins will create these fibrin threads that will create a reinforced mesh to really stop that flow or loss of blood. Looking at these coagulation factors that we have present in our blood, these are proteins that are in the plasma, they're always there, and they're circulating in an inactive state until there's damage of some sort to blood vessels or to tissues. And then the platelets or the damaged tissue cells will release the chemicals that will initiate this cascading series of events involving these coagulation proteins to create a clot. Notice that calcium and vitamin K are necessary for the clotting process, and our major source of vitamin K comes from our large intestine, from the bacteria that live in our large intestine. One of the products that they release is vitamin K, and then we can reabsorb that vitamin K into our blood, and it's necessary for us to be able to clot blood properly. So if there are conditions where we're destroying the normal bacterial flora in our large intestine, it's possible to create a symptom of a bleeding disorder where you're not able to clot blood properly. There are two different pathways that can occur with ultimately with the result of creating a blood clot. There's the extrinsic pathway and the intrinsic pathway. Extrinsic pathway is initiated when there's damage to tissue that's outside of the blood vessel, and these damaged tissue cells will release tissue factor, or what's called thromboplastin, that will then leak into the blood vessel, and that thromboplastin will start to activate coagulation factors that are in the plasma of the blood. The intrinsic pathway is initiated when there's damage to the blood vessel, and so it's not necessary to have damage to the tissue, but the blood vessel is damaged, and the exposed collagen fibers that are in the wall of the damaged blood vessel, when they come in contact with inactive factor 12, it initiates this cascade of clotting factors, ultimately that will result in the blood clot. Notice that both the extrinsic and intrinsic pathways eventually will cause an activation of factor 10. Factor 10 will catalyze the reaction of prothrombin to thrombin. Thrombin catalyzes the reaction of the protein fibrinogen to fibrin, and then factor 13 will activate fibrin to fibrin, create a fibrin clot, or ultimately the blood clot. This image will again just illustrate the two pathways to clot formation, extrinsic and intrinsic. Remember that the extrinsic pathway starts from tissue damage and the release of thromboplastin from the damaged cells, and that the intrinsic pathway begins with damage to the blood vessel wall and the exposure of the collagen fibers in the wall to inactive factor 12. And in both pathways, we will eventually end up to a common pathway where we're going to activate factor 10. Notice that we've got 13 different factors involved with this process. That it's a cascading event that once initiated will continue through the different, as long as we have all the factors in place, and ultimately we're going to convert fibrinogen to fibrin. Thrombin will be the enzyme that's going to, or the product that's going to catalyze that reaction, fibrinogen to fibrin, to create the fibrin clot. Vitamin K, notice that we have calcium necessary in these reactions, and also that vitamin K is necessary to 4 of these factors for them to be produced. We need to have vitamin K present, so with the lack of vitamin K, we're not able to build some of these factors, and in that case we can't create these pathways for the process of clotting blood. Once healing has occurred in the area of the damage and the clot formation, we want to get rid of the unwanted clots. So this is known as fibrinolysis. We're breaking down the fibrin clot, and we have an enzyme that's called plasmin that will digest the fibrin to dissolve the clot. We also have other things within plasma that is always there, and because we're constantly making small clots in our blood, that we don't want the clots to progress. So these clots can occur because of turbulence. If there's some atherosclerotic plaquing on the inside of a arterial wall, it sets up a turbulence that can possibly initiate a clotting process. So we have to have things available in our blood that are going to be always there to protect us from creating clots, unwanted clots in our blood system that could possibly block off a blood vessel. So for example, we have natural anticoagulants that are found in basophils, and we already talked about heparin as an anticoagulant released from basophils. And once basophils enter into tissues, they're known as mast cells, and they release heparin as well. Anticoagulants that people take as a medication work by preventing the coagulation factors from initiating a clot formation. So things like aspirin, Coumadin, which is also known as warfarin, used as a rat poison. These are common medications that people are put on to prevent excessive clot formation, try and prevent the possibility of a stroke or a heart attack or blockage of blood vessels in the body using these anticoagulants. Looking at just a few disorders of hemostasis that people can suffer from. Thromboembolic disorders refers to the formation of clots, undesirable clot formation. So another name for clot is a thrombus. So thrombus can occur spontaneously within blood vessels given the right conditions. And so things that can cause thrombuses, the formation of a thrombus, is generally anything that roughens the inside of the blood vessel wall, the endothelial surface. Increases the chance that platelets can get caught up in that roughness, and once they aggregate, there's a possibility of a clot being initiated. So inflammatory processes within the inside walls of our arteries, atherosclerotic plaquing, or what's known as hardening of the arteries, stasis, where you have any condition where blood is allowed to pool, for example, in the lower legs. If blood is allowed to pool or not move as quickly as it should be moving, you increase the chance of creating a thrombus or a clot. And the concern with a clot is that, first of all, it's blocking off blood flow in that area, but it also can then dislodge and now become a free-floating clot in the circulatory system. And once this has occurred, it's now known as an embolus. And an embolus can continue on until it arrives at a blood vessel where the vessel is too small for that embolus to continue to move, and now it blocks off the blood flow past that point, and you can create a stroke, or you can create a pulmonary embolism, lack of blood flow into the lungs, or a heart attack. An example of bleeding disorders are, you'll learn about hemophilia in pathology. Hemophilia are conditions where people don't have a certain clotting factor, so that they don't have factor 9 or they don't have factor 10. So if they are born without these factors, they're unable to create a clot. So these people can bleed from the simplest cut. They have no ability to stop that blood flow and are able to actually bleed to death if there's no intervention. We're going to look at blood grouping now, and there's a number of different types of systems for blood grouping, but we're looking at two of them, the ABO method and the RH. And if we just look at a definition, blood transfusion refers to the transfer of blood or some component of blood from one individual to another. And when we look at blood typing, if you are a certain type of blood, when you're receiving a transfusion, you have to get that same type of blood. Otherwise, there will be a reaction. And so why this happens, why a reaction may happen, if we look at how these groups are established, each blood type has a specific antigen on the surface of the red blood cells, the identifying flag, if you want to look at it that way. So for example, a group A type blood, type A, will have a type A antigen on the surface of its red blood cells, and a type B blood type will have a different type of antigen on the surface of the red blood cells. We also have antibodies in our plasma that will protect us from getting blood if we were to have exposure to a blood that's a different type than our blood type. So in that case, these our antibodies will bind to the antigens that are on the blood type that is not ours, and it'll cause either a clumping of that blood or a destruction of the red blood cells. We're going to use this chart to illustrate the different blood types and why there may be reactions or not have a reaction to different transfusions. So let's look at the bottom of the screen, bottom of the chart. You'll see type A. We'll start there. If you have type A blood, it means that you have your red blood cells have the type A antigen or flags on the surface, and your plasma will have anti-B antibodies. So this means that if you were to receive a transfusion that is type B blood, your anti-B antibodies will connect with those B antigens on the B blood and will cause a reaction, either a destruction of those red blood cells or a clumping of the red blood cells. If you are type B blood, it means that your red blood cells have the B antigens on the surface and your plasma will have the anti-A antibodies. If you're type AB, it means that your red blood cells have both A and B antigens on their surface, but because you have both A and B antigens on your red blood cells, your plasma will have neither anti-A nor anti-B antibodies. Because if you had either one, they would be attacking your own blood. So as a result, because there are no anti-A or anti-B antibodies in type A blood, it means that you are the universal recipient. So if you have type A blood, you can receive anybody's blood. You can receive A, you can receive B, and we'll see that you can also receive type O because type O does not have any antigens. So you are the universal recipient. You can receive anybody's blood because you have neither anti-A or anti-B antibodies. If you are type O blood type has neither A or B antigens on the surface of the red blood cell, but their plasma will have anti-A and anti-B antibodies. So because their red blood cells don't have A or B antigens, type O is known as the universal donor. They can give their red blood cells to anybody, and because there are no antigens on their red blood cells, it doesn't matter whether you have anti-A or anti-B antibodies in your plasma, they're not going to attack those O red blood cells. So O is the universal donor, and type AB is the universal recipient. Just to illustrate what may happen if you give or receive the improper blood type in a donor-recipient situation. So if type A blood donor gives his blood to somebody who has a type A blood, so that person will have the anti-B antibodies in their blood, but the anti-B antibodies will not connect with the type A antigens on the red blood cell surface, and so there will be no agglutination. So the red blood cells will not be clumping together. If you look at the second example, if a type A blood donor gives his blood to someone who has type B blood, that person with type B blood will have anti-A antibodies in their plasma, and those anti-A antibodies now will connect with those type A antigens and will cause an agglutination or a clumping reaction where these red blood cells will all clump and agglutinate together, and obviously that's not a good thing. The Rh blood grouping refers to another type of antigen found on the surface of red blood cells, and this is the Rh antigen. And this antigen was first discovered in the rhesus monkeys, so this is where the name comes from, Rh, the first two letters, and rhesus. And so this is an antigen that on 85% of North American population, we have this Rh antigen on the red blood cell surface, and if you have that, you're Rh positive. If you don't have the Rh antigen on your red blood cell membranes, then you're Rh negative. So when we're looking at creating a blood type, We're using the ABO and the RH antigens to determine what blood type you are. So if you are a type A blood type and you have the RH antigen, then you're A positive. So the positive or negative refers to the RH antigen. So if you're an A blood type with no RH antigen, then you're A negative. And that's the same for the B or the O or the AB blood groups. The last thing we're going to look at is a condition called erythroblastosis fetalis, or hemolytic disease of the newborn. And this can occur when the pregnant mother is determined as RH negative blood type. And if her first child is RH positive, and if there's any exposure to the fetus's blood, the RH positive blood, so during the birthing process, for example, and there's bleeding, and if there's any chance that the RH positive fetus, the blood from the fetus is in contact with the mother's blood, the mother will then create, her blood will create anti-RH antibodies. So this becomes a concern if the woman becomes pregnant again, because if she becomes pregnant again with another RH positive fetus, and so recall that it's 85% of the North American population are RH positive, so it's a much higher odds that you will have an RH positive fetus. And in this case, with the second RH positive fetus, now the mother has in her blood anti-RH antibodies that can now enter through the placental circulation, attack the blood of the second fetus, and cause a fatal reaction to that fetus.